So a summary for this part is that PCA is a linear method, so keep that in mind. Always use log scale data. Always use the scale data or use the PCA, and that is already in the exercises as well, how you make sure that you're using z-score data. And always transform your data, make sure it's in log scale or linear log. So PCA is an interpretable dimensionality reduction, because if, as I mentioned before, in principal component 1, you can measure what is the importance of those genes to separate your samples. The same thing for principal component 2. There is a meaning behind them, of those principal components, such as principal component 1 is the one that stores the highest variance in the data, and then they are sorted in that order. And then, in many cases, but not probably not much for bulk RNA sequencing, but for other methods, you can use that to filter variance in your data and use only the top principal components. So so for example, in our exercises later on, we'll be looking at the top 4 principal components because there are not that many samples in your data. But then you'll see and explore a little bit that on the exercises later.